case study, so I think maybe a lot of people did not read the case study. But that's okay. We'll walk through a little bit of the HSV case study as well, and talk about that a little bit. And then there's no class on Thursday.
Obviously.
And then
It's Easter break.
class on Monday.
Oh God, where am I?
See you guys
Where's my
again
mind?
until
Thursday.
so like a week and a half. Is it really Easter break?
Yeah.
Okay,
Yeah.
I do see you on
Tuesday,
okay,
Monday.
yeah,
This
sorry.
week.
Okay, Tuesday is Monday.
Oh, sorry, Tuesday.
Golly.
Good Thursday.
I don't like
Friday.
this Monday classes on Tuesday.
If y'all are here like 5 minutes and waiting on me, somebody come get me because I'm not going to.
I'll come get you.
Prep day for your presentations.
Oh yeah.
What day is the rest? That is Tuesday.
25th?
Yeah, it's pretty
Yeah,
good.
the
We
25th.
have the 23rd to go.
Yeah.
I'm
Slacker.
No, she doesn't believe in this academic Monday, which I don't either.
Tuesday's 23rd. So that is your prep day for your presentation. Presentations are Thursday. Okay, so let me tell you about prep day for presentations. Yes. Let me tell you about prep day for presentations. I will be here to provide feedback to any group that wants my feedback. If your group is confident that you do not want my feedback, then that is the choice that your group makes as to whether or not you are here that day to gather my feedback. Hint, hint.
I want your feedback.
Read between the lines there. If your group wants to practice your presentation in here that day, we can work that out. You know, we can have certain groups coming in at different times. If you'll let me know this
You
week
want to do that?
sort of what your
I have
group
more
wants
time
to
this
do
week.
and
After to work on the
how we want to do that, then I'll kind of set up times for
Yeah,
people
that's
to
fine.
come, so you're not here the whole time. So I'll say this group comes the first 15 minutes, the next group will come the next 15 minutes, so forth. So let me know, shoot me an email this week, let me know what your group wants to do in terms of getting feedback. So that is
Tuesday.
Tuesday, the
I'm
23rd.
sending you off to class.
And then presentations will start Thursday, the 25th.
Is there gonna be like a list on who's going first by group, or do we just raise our hands like who wants to go first?
Let's figure that out right now, actually. Because I think some of you have conflicts with those days. I think I've heard from a couple of you.
Do you have conflicts? I haven't looked.
Not really.
I don't know if Thomas has conflicts or not.
You know
Okay,
who's here.
so we have presentations. I think we can probably get them all done in 2 days, but let me see. So we have the 25th, the 29th, and the 2nd for presentations.
Yeah, go another.
When you wanna go?
Whatever. I'm not picky.
Alright, does anybody have a desire to go on the 25th?
Yep, I'm saying 25th.
Get it out of the
You
way.
have to go on the 25th? Oh, the 29th, you want to be here. Okay, that's right, you were the one.
Easier to get it out of the way.
Okay, so
Understand.
your group can definitely go on the 25th.
I'll say.
Leah, your group wants to go on the 25th.
Yeah, why
See.
not?
Anybody else have a desire to go on the 25th? Anybody else have a desire to go on the 29th? What was the other, is somebody else raising their hand? No, it's not.
Hey, someone.
Okay, so we have this group, and Megan, we have your group. 25th or 29th? You really want the 2nd?
We did 29. They don't get a pick, so.
Huh?
We'll go on the 29th.
29th. Alright, this group's gonna be the 20th. None of them are here to defend themselves.
Nope.
Alright, so 25th, I have Nathan's group, I have Sophie's group. You guys on the 25th. I have Leah's group on the 25th. The 29th, I have Catherine, Alessandra's group, I have Tally's group, and I have Megan's group. We're missing this group. They're going on the 25th. That's all the groups I have. I'm missing a group over here. That's it. Alright. general overview. What did they do in this paper? Okay, yeah, give it a shot.
So basically, what they did in this paper is they used vaccine candidates to determine the efficiency of potential vaccines to combat the aerosol RVFV virus, which is the Rift Valley fever virus. For two, it was because if that virus is in the form of aerosolization, it has been considered as a bioweapon, and so that's why they're producing or trying to find out which vaccine is effective to combat this potential weapon.
Okay. So they're basically developing a vaccine for Rift Valley fever virus, RVFV, which could potentially be used as a bioterrorist agent or as a bioweapon because of its ability to spread through aerosol, through sprays, through sneezes, or any sort of spray that we could use to distribute this virus. Anybody have anything you would add to that? So, what did they do? How did they do this? No, somebody other than
Okay,
me.
yeah, because I'm like.
Alright, why don't you guys do this? Why
It's on
don't you
campus.
take 10 minutes,
This one.
talk in your groups, go through the questions,
Yeah, it's on campus.
prepare
No,
your
go back.
answers. Megan, you can join another group if you'd like.
My bad. I'll
Read
do
through
it.
the questions, talk it out in your groups, and then we'll report back.
Give me a minute.
Which question was it? Question was
What they did.
what did they do?
Like, how did they do their experiment?
Oh, how to do it? They use ELISA
What
to
did they do? Like, why, or how did
This
they
is
Did-
more like
Let
experimental.
me read through it again.
Oh, it's more
Go through the questions on the
I
worksheet.
completely read the wrong
Okay.
one.
Describe the technique that they use. It was indirect ELISA. And they were looking at specific proteins and genes, plasmids. One was GNC3D. One was C3D, NP12, all that stuff.
I think those were the different vaccines.
I thought that they created those vaccines because they isolated those specific I don't want to say things, but they specifically isolated these components to put them into a vaccine on its own. At least that's what I thought when I was reading it.
I thought what they did was they took different types of vaccines, injected different groups of mice, exposed them to the virus, and then the ELISA was to measure the immune response
Yeah.
to the virus, which would change because of the different vaccines used.
Okay.
That's what I think. That's what, and because when you get to figure 2 or 3 survival rates, and then how
Yep.
sick they got. So I think those proteins, I think the plasmids was the different types of vaccines.
Okay. Yeah. Which I was actually really grateful for molecular. I could actually understand the figures.
Yeah.
So I think that's a plus to taking this class. But for figure 2 and figure 3. So figure 2 was it monitored the mortality rates of the mice. Figure 3 was more so the clinical symptoms of mice, and I could have like how in molecular go through each one and say what the outcome was, but overall it was a GNC3D that had the most effect on protecting the mice from the aerosolization of the RFVF. Cool. And then a live attenuated vaccine. That's kind of like the Flonase. It's still a live viral particle, but it's like reduced in its like
Pathogenicity.
yeah, but it can still become virulent if given like proper conditions and stuff. For number 6, I didn't understand like what she meant by what would different vaccines have different effects on different animal models. Did that have to do with like the antibodies and the symptoms?
I said different animals might have different immune systems.
Okay.
Like different antigens, or different types of vaccines, like different
Responses.
Okay.
Maybe
All right.
it's
So
maybe it
pretty
doesn't
straightforward.
respond at all.
Okay.
Yeah.
Okay. I guess that's just pretty straightforward. And then for 7, I was like, they should study the GNC3D DNA and the MP12 further. And perhaps like place them both into a single vaccine, like the MMR vaccine. That's what I think that they should do as a further step after the research. That's what I think.
Come on, please.
You guys, I
Just
said
the first letter.
increase like stressful living before and during and after. It's like a bioterror attack. You know,
Got your text.
it's something.
True.
Could be anything.
True. That is very true.
Yeah.
All right, we're done.
Yeah, right.
We're done. Nice job, everybody.
Yeah, like
I'm sorry I could not have been more help. I picked the completely wrong case.
It is fine.
And I just now realized the title's right there. I did not.
I did the same thing. The table for case study.
Yeah, the other one that's more like the HSV. Yes, I read that one. I did not think to connect.
It is fine. It is fine.
And yesterday she was like, I've been looking at the
Plus, I go into the files
So
of the case
I'm like,
studies,
Yeah.
and I didn't find it there. I just found that one.
No,
And
it's
that's
just-
why I
It's
assumed.
just
Under file.
Yeah.
Yeah, it's just under files, not under case studies.
It is fine. It is.
This is why we have Leah.
And Evan. And Evan.
yeah.
It is.
Teaching then,
It is all fine.
And at this table, we love and appreciate Evan.
Yes. The champ.
Yeah.
The champ.
I'm gonna not look at the same. Go back to this.
Okay. And I wanted to visit to get that house.
So let's talk about, I think you and Paul were doing that though.
Yeah. 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 I emailed them. For Friday.
I was called.
Yeah.
Okay. Guys had a chance to talk about some questions?
Mm-hmm.
Okay. So let's talk about this a little bit more. All right. So we know they're looking at vaccine candidates for this particular virus, which can be pretty bad, particularly in the aerosol form. Somebody tell me about RVFV first off. Like, what kind of virus is it? What sort of symptoms does it cause? What do we know about it? What do we know about RVFV? What kind of symptoms does it cause? How is it spread? Yes. Okay, so we know it is transmitted through mosquito bites. Okay, that's typically how we see it. What else?
Outbreaks in like long-term experiments, which is why they've been walking towards that one to the
Okay, so there's certainly been outbreaks that they've studied that led to this thinking of we need to be better protected against the aerosol spread of this type of virus. We know that it's characterized, so this particular Rift Valley fever virus causes Rift Valley fever. So this is a case of a virus sort of causing a disease that we've talked about a couple of times during the semester. We know there can be widespread outbreaks. So when it is transmitted, there usually is not good protection against it. It causes neonatal mortality in animals, so neonates do not survive, and it causes flu-like symptoms with occasional complications in humans. We know that mosquito bites is the primary form, but we know that particularly when we're talking about human infections, aerosol is a much more sort of dangerous way that things can be spread and something that we need to be better protected against. One complication with Rift Valley fever virus is that it has a pretty long half-life. It says that to remain viable for more than an hour under optimal conditions, a half-life more than 77 minutes at 25 degrees Celsius and 30% relative humidity. So that's a pretty long half-life for a virus. So that means if I were to blow some Rift Valley fever virus on this table and come back 77 minutes later, half of those virus particles would still be viable and could still cause an infection. So that is a particular concern when you're talking about an aerosol virus, because we can spray it into the air or spray it through the ventilation systems or something like that, and it's gonna linger and not just affect the people in this room right now, but the people in this room in the next class could also be targeted or be affected by the virus. So that's an interesting sort of thing to think about with these sort of viruses that are tools of that can be bioweapons, is how long do the viruses hang around. And that's something that, as a general rule, we don't really have to worry too much about when we talk about general spread of, say, the cold virus or the flu virus or things like that. Typically those viruses do not live long or do not survive, remain viable outside of a host cell very long. So that's a little bit of a unique thing to Rift Valley fever virus. Okay, so they're developing these vaccines against particularly the aerosol form of these viruses. Why is this a concern now? Sophie, you kind of hinted at this.
Sorry.
So why are they now looking at preparing these vaccines?
Because with the outbreaks of the aerosol, they assuming that's the only way they thought it through the aerosol infection from the labs.
Okay.
So because of that, then I'm thinking, oh, if this is happening here, then when someone could use it, then everybody would do it.
Okay. And why is it important to think about? So we know that we have a way to protect in mice. They said this in mice. Protect against an intraperitoneal infection. So that's when they injected the mice with the virus, and they have pretty good, a pretty decent vaccine against that sort of Rift Valley fever virus. Why do you think it would matter if it is the aerosol form of the virus? Why can't we just use the same vaccine?
Because evidently, like, the current vaccine that they have that's not for the air form of it is not as effective to protect people and animals.
Why? Yes. That's right. So we have different entry points, right? We talked about sort of some different points of entry that viruses like to use. So obviously an intraperitoneal injection is going directly into the bloodstream, whereas this virus in the aerosol form, we're breathing it in, so probably it's coming in through our respiratory tract. Maybe we're touching something and then touching our mouth or touching our face or something like that, so it's coming in through one of those sort of entry points. So the point of entry is different, therefore the way that we target this virus, to attack this virus, would be different, and therefore the types of antibodies, the type of immunity that we need to protect against this virus is going to be different. So viruses with different entry points are going to have different, everything about it is going to be different. Okay, so we got to develop this other virus. Okay, so let's look at figure 1. It says describe the technique used to obtain the data in figure 1. So somebody tell me a little bit about figure 1. What were we looking at? What type of technique did we do first? Okay, they did an ELISA. So we talked about ELISA, the way that we use ELISA as a tool for looking at viruses. So that was way back in chapter 1, where we were able to test for the presence of a specific virus by testing for the antibodies to see if the antibodies were present. So somebody tell me what their data shows in terms of figure 1. Oh, let me scroll down, sorry. So if you look at the data, we did an indirect ELISA measuring RVFB specific IgG response in mice immunized with indicated vaccine regimen. So that's 1A. So what does this data tell us in 1A?
And the NP12 vaccine produced the greatest IgG response.
Yep. So the NP12 vaccine has the highest sort of endpoint titer. So that tells us we had the most antibodies produced when we applied the NP12 vaccine. What is the NP12 vaccine?
It's the live attenuated one.
That's the live attenuated one. And then the other ones are different combinations that they have produced of this GN. What's GN? That's the glycoprotein from the virus. And so we've combined it with various different things. So we either have it alone, we have it in combination with the C3D, we have it with the Reb, and then we have the C3D Rep combo, sort of the two together. Those two together is the third one. So these are the different vaccine candidates that they are testing to see what we can get. Obviously, the live attenuated produced the best immune response. What about GN alone?
It's not as effective.
So GN alone is the first line, so that was the lowest, produced the lowest antibody response. And then the other three were somewhere in the middle, like sort of all sort of comparable, the other three responses. All right, so let's look at B, figure 1B. It says, this is an indirect ELISA measuring RVFB specific IgA responses in mice immunized with indicated vaccine regimen. So what sort of, so they're still doing an ELISA, right? They measured, what have they measured here? We have an OD at 405 nanometers. What does that mean?
Absorbance?
Absorbance, right? They measured absorbance, so they looked for a color change and on their ELISA plate, they put it on their plate reader or whatever they have, they measured the absorbance at 405 nanometers. So the higher the absorbance, what does that tell us?
The more concentrated of the more present the antibody is.
That's
Yeah,
right. So the higher the absorbance, that means we had a darker color change. That means we had more antibodies present in our sample. So which ones here did well?
GN and
GN or GNC3D. So those two produced the most IgA antibodies in response to the RVFB. The other three did not produce much. All right, and then C, what were we looking at in C? So this says neutralizing antibody response in mice immunized with indicated vaccine regimens. This is 1C. So what are we looking at here? Are they doing, is it still ELISA? Yes, it's still ELISA. So now we're looking at the presence of neutralizing antibodies. So here we can see that GN really did not produce much of a titer of neutralizing antibodies. The other 4 did pretty well, with still MP12 kind of up there high, and the rest of them similarly. So the lines over the top with the stars, what do you think those mean?
It's
Significant difference. So that's their statistics, which as biologists we hate that word typically. We don't like to think about statistics, but we do from time to time. So all of those lines with the stars, that's telling you whether it's statistically significant. So that's their t-test that they did. They got their p-value. You guys all know that from Dr. Hewitt.
actually from Sprinkle.
What?
From Sprinkle.
Oh, from Sprinkle. Okay. All right. So this is the first indicator to tell us kind of how these vaccines will do in terms of antibody production, testing and ELISA. All right. So let's look at 2 and 3, figures 2 and 3. All right. So I want. All right. How about Evan? Your table take figure 2. Kelly, your table take figure 3, and sort of talk about those figures, be able to explain those figures, and then you're gonna sort of battle it out to see what are the differences between these 2 figures, and talk about what the data shows. And then you guys, I want you to think about number 7, question number 7. Where it says, think of one experiment you could do to further their results. What is
There to monitor morality, the mortality of the mice. The outcomes for it are as shown given how the vaccine progressed in these mice, and everything but everything but the GN. Also, so everything but the GNC3D, the mice did poorly, except for when the GNC3D was administered, they had 100% survival rate compared to the Rep GN or just GN or the other one.
The other 3 all end up with a 40% survival rate.
Which one did?
2A?
Yeah.
All of them end up with a 40%.
Yeah.
That's true. That one, yeah.
And then the differences between figure 2 and figure 3. One dealt with mortality, one dealt with the symptoms, and it's not really it's a huge difference, but because the GNC3D did so well with its survival rate, it also had 0 cases of symptoms in figure 3.
Yeah, which is
good. But it seems too good to be true, but never know.
they even get infected.
So you never know. But yeah. Is that the only difference that
Well, when you look at the symptoms, you'd want to look at the length and how bad they get. And some of them are
pretty
straight
bad.
up.
I think that was Rep GN that just skyrocketed after a couple of days. Yeah, it was Rep GN that skyrocketed, along with just GN. They were pretty high up, but Rep GN but it's a correlation because all 3 of them, besides the GNC3D, they started off as 100, but then they slowly dipped down to 40%. Also had a higher, symptoms, higher clinical symptoms. Cool. That sounds good.
Oh, the blood rushed to my head for a second.
Hmm.
I don't want to do it.
Too bad.
I'm lazy.
How's your weekend, Thomas?
Like studying physics.
Sorry to hear that.
Did you look over the Kahoot questions? Did you look over the Kahoot questions?
Those are going to be on the multiple
There
choice.
was the
I
one we did in
remember.
class, and then there's another one that actually we didn't go over that was based more on the videos that we watched before the special quantums.
The Kahoot questions were the multiple choice questions.
For everything.
I just went through it. I just went through the game.
The Kahoot questions was the multiple choice questions.
Well, good thing I went over it.
That's what he did on our exam for last year.
We memorized the rap.
Yeah, legit. Memorize the rap. I forget it now, but I did memorize it.
I don't know it very well. But
So cheesy.
I like that.
Oh
It was
my
so
god,
easy.
it's just like, oh.
You're trying so hard.
So cheesy.
I liked it. Okay. I liked it.
So cheesy.
So basically all these,
Yeah, all the what every single one, but like
but most.
he chose like random ones, and they were the multiple choice questions. So I mean, I didn't really study that much for the Kahoot. Like I didn't
Yeah.
study off of like anything else for the multiple choice but the Kahoot.
Okay, so let's start with figure 2. Do you guys want to explain to us what figure 2 shows in a nutshell?
I've been talking too much. Why don't you three do it? I've been talking too much.
The top picture shows that the mice who were vaccinated with GNC3D, they all survived, compared with the rest of them that all went down to a 40% survival rate within the 2 weeks, which is kind of similar to all of the controls in the other.
Okay, let's talk about A and then we'll go to B. So this is looking at survival rate. And so this is percent survival. On your x-axis you have days post aerosol challenge. So these were mice that were exposed to RVFV and vaccinated with the appropriate vaccine. So, and then determined how many days they survived following exposure. So the kind of brownish circles, those are our GNC3D vaccines. You can see they sort of maintain 100% about 14, 15 days post-exposure, whereas everything else in some manner eventually dropped to right at 40% post 2 weeks exposure. So that tells us this vaccine is doing something to better protect our mice against our RVFV than the other vaccines. What's the first thing that you think it's probably doing to better protect us based on the data from figure 1?
It is administering it's basically causing an immune response.
It's causing an immune response. What did figure 1 tell us about the antibody production?
More antibodies.
We're producing more antibodies with this vaccine than some of the other vaccines. We're better able to fight off this infection. All right, what about 2B?
Yeah.
Oh.
Obviously wasn't as good as 2A, but they compared the controls with the live attenuated one, and then the naive one, which those died, and then the 2. The live one stopped at 40%, and the DNA control also stopped at 40%. So the other 2 died. So it's kind of like the control and the these were at 40%. Makes it kind of hard to tease apart how good it really was.
So what is figure 2B? What are they actually testing there? Are they testing the effectiveness of the GNC3D vaccine?
Aren't they testing the mortality rate?
Okay, so they are testing mortality rate, but what are they giving the mice?
What are they giving the
Either the
What-
live attenuated one,
Yeah.
or the controls is just like the plasma without any.
That's right. So they're really testing not necessarily the effectiveness of this specific vaccine, the GNC3D, they're testing the effectiveness of a DNA vaccine versus the live attenuated vaccine, the MP12. Okay? And they're testing the effectiveness of the plasmid in general, right? So maybe the plasmid itself, without even anything in it, has some sort of protective feature or something. Did it?
Nope.
No, it did not. Like, so, which one is that? That's the
Direct control.
that's the open squares, which
All DNA control.
so it dropped to about 40%, right?
Mm-hmm.
Okay, so I think that's the DNA control. After about 2 weeks, only about 40% of those mice survived. If we look at our representative control, so that's just a complete control that had no protection whatsoever. All of those mice died. Our NP12, which is our live attenuated vaccine, also sort of eventually hit this 40% mark at about 2 weeks. So this is telling us that our DNA vaccine has the potential to be as effective, at least, as our live attenuated vaccine. Okay, questions about that one? All right, figure 3. You guys want to talk to us about figure 3?
So 3A shows the sickness score with the days post aerosol challenge. The figure A has the vaccine, so
Sorry.
Oh yeah, that's so much better.
yeah.
Yeah.
The GNC3D, the mice that were treated with that one, they had a 0 sickness score 2 weeks post the aerosol, so they were consistently 0. And then with the GN vaccine, it was around day 4 post aerosol challenge that they started to develop clinical symptoms, and then eventually went back down to a 0 around day 14. And then with the REC-GN and then the GNC3D/REC-GN, it was about 6 days post challenge that they developed symptoms, and the GNC3D and the REC-GN eventually went back down to 0, like the GN did, but the REC-GN stayed about like 0.5 on that scale. And then the figure B shows the controls. So again, the DNA control is about 3 days post that it developed the symptoms, and then it never fully went back down to 0. The MP12 was about 6 days post, and then eventually went back down to 0 around day 13. And then the other two, they like shoot up and stay up. They don't even kind of tilt back down, so they hit the maximum stop.
Okay, so this one is looking at a sickness score, and you can read they define what they assessed in terms of their sickness scores for their animals. So this is basically looking at to say, when did these mice start developing symptoms post